It's what I do. Okay, so now that I'm recording, I can just start in on marine ecosystems once again and say that they can be defined by a variety of different parameters. And I think the way we'll just do it for this lecture is we can say that we can define it by the proximity to the coast. So there is what we call open ocean or the pelagic zone. And really this is defined by essentially low nutrient levels. So really regions like Sargasso Sea and the Pacific gyres, they're extremely nutrient deficient. So organisms that live there are well adapted to those conditions. I'll get into that. But then of course, as you go in towards the coastal regions, you have different factors like upwellings of currents, runoff from terrestrial ecosystems. So often these are more nutrient rich. And then of course, there's also like, as you get into the coast, you have the continental shelves, and the further out you get more of the abyssal zones and the deeper zones. And then with that, this is also where the light comes in, because you have euphotic zones. So this is where the sunlight can reach into the water. This is typically about 200 meters. And then once you start getting below that, there are different ways of dividing that up. But the simplest way is just to call it the aphotic zones. So you don't really have any light getting into those regions. So organisms that are there have to derive their energy and primary production energy from another source. And interestingly, though, I mean, you know, oceans, they're these gigantic bodies of water, and there's wind and there's currents. But the thing about surface waters is that they can be a bit more isolated. I mean, you might have wind currents that are mixing up the water a little bit, but when researchers go out into the ocean and they look at different organisms that live in different regions, you get these geographical, like isolated types that live in different regions. So that differs a bit from when you have deeper in the ocean, where there are a lot of more large convection currents. Those tend to be maybe a bit more mixed, but this global ocean conveyor belt, it takes, from what I read, it's 1 cubic meter of water will travel through the whole conveyor belt in about 1,000 years. So it's not like it mixes that fast. So as you're traveling through that, you could still wind up having conditions changing slow enough where things that weren't adapted there die off, and then other ones might move in. But either whose, you can have these different areas that are defined by these parameters. If we start in the euphotic zone, this really ocean-wide is the main region for primary production. And maybe if you're not familiar with what I mean by primary production, it's basically you're synthesizing organic molecules into, or rather you're synthesizing organic molecules from carbon dioxide. So it's basically photosynthesis. And then you have molecules, then you have biomass, you have carbon that can be utilized by other organisms, heterotrophs. And ocean-wide, the predominant primary producers really are, it's actually 2 species of bacteria that are photosynthetic. So you have one species, Prochlorococcus, and another Synechococcus. And these guys actually account for 5% of the global all photosynthesis on the planet. And if you think about the amount of forests and the amount of terrestrial photosynthesis, that's a substantial amount for 2 species. And yeah, there are plenty of other organisms in the ocean that also photosynthesize, but these guys take up 1/4 of that. But as I said about the different marine habitats, that especially at the surface water, they can be fairly isolated from each other, even though, yeah, they're continuous bodies of water. So you actually have different variants, like what you call ecotypes of these 2 different species that are going to be found depending on how sensitive they are to light. Some will actually live in lower parts of the water column, some in higher parts of the water column. Some will be in higher latitudes and some in lower latitudes. And I got something that's really neat, because the way that they've been studying these organisms is you go out into the ocean and you can measure all these different parameters, the temperature, nutrient conditions, any other parameters, and then you can see where they particularly like to be. Light, of course. And what they've done is they've actually modeled this on a global level. This is from MIT. And if we play it, what they did is they are actually looking at oceanic conditions over the span of 4 years. And then looking at essentially 4 groups of species, 2 of which are our primary producers. And you can actually look at over 4 years how the different populations of the groups are changing depending on season. Is of course, as you start getting into summer seasons, you're going to have warmer ocean temperatures, and then you're going to have increases or decreases of the 2 different species. So you could actually see as you approach, they didn't unfortunately have the seasonality up here, but as that red increases, that's when you start seeing more winter accretion. That's the diatoms towards the Antarctic. But then you also see shifting patterns. Here's the blue coming up in, the yellow and the blue are actually the Synechococcus and the flagellates are sort of changing around depending on the season, dominating at different times of the year. And you can see these fluctuations of the populations over time, which is just fascinating that you can actually do this on a global level. The models behind this are extremely complicated, but what they're essentially doing is, after studying, in particular, these 2 organisms, they can get an idea as to when you would expect them to dominate and not. So it's measuring in ecology what you call their functional space. The conditions that really are optimized, that they are optimized to, rather. Let's see, there we go, up. So then with this primary production, you have carbon that's being produced by photosynthesis, and this enters, and this is very much a marine term in microbial ecology, what they call the microbial loop, or it's been a bit more updated, it's called the microbial food web. This was in one of the papers that I assigned for the reading from the previous module. But the idea is that you have not only the photosynthetic cells, but also the heterotrophs that are eating up all the carbon that's being produced by these cells. And they're being grazed upon by eukaryotic microorganisms like protozoans, the ciliates that we see swimming around. And then they're being pooed out, and then you have dissolved organic carbon. And it's not like larger organisms can just go in and absorb dissolved carbon. You need to have that taken in by bacteria, and then organisms eat that, bigger organisms eat that, and then it becomes a part of the food web. So they call it this microbial loop. And also you have viruses that are viruses are everywhere, as we know, but viruses play an important part, because when they go to infect bacterial cells, they'll burst open, and then all the dissolved organic matter can also be released and then be eaten up by heterotrophic bacteria. So this microbial loop here, this is really where you see the microorganisms that are driving essentially the food web. So they're allowing what would otherwise be just dissolved carbon to go back into the overall food web, or they're driving that. And another really interesting aspect about marine ecosystems, maybe people have heard this term, marine snow. So it's this, when you have all this carbon and all these dead particulates, dead cellular particulates, you have fecal matter coming from all different macroorganisms. What you wind up getting is this accumulation of organic matter into these small particles that are also inhabited by different microorganisms that are feeding off of that carbon, as well as other elements that are in the marine environment. So you get this sort of food web that is essentially on all these small little particles. I mean, you can have, of course, free living cells in the aquatic environment, in the ocean, but you get a lot of them that are also driving carbon flow that are inhabiting this marine snow. And it can be quite interesting because there was one study that was done showing that I forget the volume of the area that they were looking at. It was off the coast of Australia, but they were looking at different marine snow particles. And you could actually find that they had slightly different communities on each of them. So you can get this biogeography on marine snow on each individual little particle. And of course, yeah, a lot of that has to do with the types of carbon that are conglomerated into that. But it also has to do with how the different types of organisms, how much they can disperse. So their ability to swim around, their ability to colonize, and whether they colonize that particle first. So in ecology, you call that a founder effect. If you get there first, maybe another organism can live there just fine, but you got there first and you can outcompete that. So you get biogeography even on marine snow. And of course, marine snow, it'll precipitate down, and this is what's forming the ocean sediment. So you get this ooze of organic matter that's at the bottom, and that's really where the majority of microbial life is living, at the bottom. As I recall correctly, the figure was, I think it's 66% of all bacterial or prokaryotic cells are in marine sediment. I mean, obviously because it's such a large area, but this is what you really get. This is what forms that sediment, is this marine snow that just drops down. Again, the thing about marine ecosystems and cells that live there is, especially in the pelagic zones, the cells actually are very, very small. For example, your typical bacterial cell volume, if you're looking at like a pathogenic bacteria or another disease-causing bacteria, or even just one that you find that could be anywhere. The average range tends to be, when you look at cell volume, it might be between 0.4 and 3 cubic micrometers. But Prochlorococcus, its maximum is 0.2, so that's the photosynthetic one. And there's another bacteria called Pelagibacter, and it's actually 0.01 micrometer, and that's very, very small. It's actually the smallest bacterial cell that we know of. Maybe I can shout out, why are they so small in these conditions? Why would they? What advantage do they have in being so small? Anyone, just yell out. I guess it would have something to do with the nutrient concentrations, right? Absolutely. They're adapted to low nutrient concentrations. They can only grow so large because there's only so much material they can grow with. And it takes quite a bit of energy to produce proteins and other macromolecules if you're going to have more complex functions and you're going to grow to a larger cell size. But despite being so small, this is the most abundant. The Pelagibacter is actually the most abundant organism in the ocean. And during warm seasons, it can actually make up 50% of all cells that you find in the ocean. That's why it's actually named Pelagibacter ubique. It's ubiquitous, it's everywhere. So it's quite interesting. And this organism wasn't discovered until I believe it was the mid-90s, late 90s, early 2000s, because there was no way to culture it. There was no way to understand its physiology. It was only when we had DNA-based methods that we really understood what it was and how abundant it was. And then once you find something, you start developing more techniques to go in and study it further. And then maybe just a little bit about some of the extreme environments that you have in marine ecosystems. And I think the most famous, of course, is going to be these black smokers and volcanic vents. So these are vents that are much more in like volcanic ridges, mid-ocean ridges, in the abyssal plain. And here, I mean, these are driven by volcanic activity. So you have water that's seeping into the crust, being heated up very, very hot. But because they're in the abyssal plain, there's so much pressure that you can actually get water that is superheated. Some of the vents are spewing out water that's 400 degrees centigrade. So when marine biologists go down to try to sample from these things, they can't get too close, otherwise their instruments melt, literally. That being said, the thing about these black smokers that's fascinating is that it's not just hot water that they're spewing out, it's tons of minerals that the water has basically dissolved into it and then being spewed out. So you got a lot of different minerals that are available to the microorganisms. And of course, it's not like all the water is 400 degrees all over the place. There's a temperature gradient. So the organisms that live there, you'll have a gradient of microorganisms that can live quite close. There are several species that can live at 121 degrees centigrade. And then the further away you get different varieties of species. But the thing about this particular ecosystem is that there's of course no light. So there's no primary production that's being driven by light. It's being driven by chemistry. So the minerals that are coming out of these volcanic vents, a lot of them are quite reduced. And by reduced, it means that they're electron rich. They have a lot of electrons. And if you have something that's electron rich, then you have something that can suck up electrons, it's electron hungry, you have energy. And then you will always find organisms that will be able to utilize that energy potential. So the organisms that live here, a lot of them are using reduced inorganic sulfur compounds like hydrogen sulfide, the rotten egg smell. There's also a lot of reduced metals that are coming out of these black smokers. And now that you have microbes that are utilizing these minerals, you can get other organisms further up the food chain that can feed off of these. So you get these giant worms, these tube worms that I'm sure quite familiar, people have seen them. But the thing about these tube worms is that they form a symbiose with the bacteria. So this is an example up here. So you have the tube worm, but inside you'll have all the different sulfur reducing bacteria that live in it. So as the minerals come in, so the yellow part is the symbiont, the bacterial symbiont, and the orange part here, the pink part, is the worm. So you'll have minerals coming in. The symbiont is utilizing that and fixing carbon dioxide and producing biomolecules that the organism, that the worm can then use to produce and basically feed off of and build up its own biomass in a heterotrophic form. And there's a variety of other functions that can be shared between two organisms so that they can essentially, they've adapted to this rather extreme environment. And I'm actually going to save, I was going to put in a little extra about this, but this lecture is probably going to be a bit long anyway. And I have a lecture I want to give on the last course days about these ecosystems are typically thought of as being really good models for studying the origin of life. Because this is where most scientists kind of concur that if life started anywhere on this planet, it was probably here 3.5 billion years ago. So when you're looking for extraterrestrial life as well on, say, moons around Saturn, like Enceladus or Europa, and you see these geysers of water spewing out of Enceladus, which was found a few years back, that's what they're thinking of. So I just find that fascinating, but we'll get into that later. Then maybe we can also go a bit into the differences here. Now we have freshwater lake ecosystems, and really one of the main differences, and this is very, very general, but one of the main differences is that in freshwater systems, you have a much greater effect of the seasonality. So as you start getting into summer, you start seeing a greater degree of stratification. The surface water is warming up, it's separating out from the cooler water below, and then you get what's called a thermocline. So you get this gradient of temperature and the water is stratified. Everyone's jumped into a lake and it's been just fine at the surface, and then you start freezing once you go a couple meters deep. At least I do. I'm awful when it comes to cold water swimming. I just don't do it. But this changes as you go into winter because you start having the surface water cooling, then you start getting a greater degree of mixing. So you have upper layers that were warm before, and now they're mixing with the lower layers. And this is where you start getting nutrients being mixed in, because as you have water flowing in from land, you have different nutrients that are coming into the water, start sinking down, because as it's going in, it'll drop. But over the wintertime, you start getting it stirred up. I mean, when you see the water in the springtime, it's cloudy, right? It's muddy. And then it starts to clear out over summer. So this is what you're seeing, is that mixing. You can have nutrients being transferred between these different areas by diffusion, but it's much, much slower. But this is the thing about the nutrient mixing, is that this is what's determining what's called biochemical oxygen demand. So when you have microbes that are living, and it's mainly microbes that are determining this, the degree to which nutrients are being mixed and the amount of how fast they can grow is determining how much oxygen is going to be taken out of the water. So if there's a large nutrient influx, you wind up seeing a huge bloom of organisms, and people have seen this before. I mean, as in you guys have probably seen this kind of idea of nutrients coming in, eutrophication, and then you see dead fish. So the biochemical oxygen demand becomes extremely high, because yeah, you'll have photosynthesis going on at the top because there's a lot of green stuff, but there's a lot of other organisms below that that are eating up the carbon. So as you get further down, the oxygen demand is extremely high. And then you start seeing these dead zones. That's why you see dead zones in the Baltic. So in lake systems in particular, this also applies to, like I said, this applies to, oceanic systems and sea systems, but you see this very prominently in lake systems. But also as you go further down, when you have this gradient of oxygen where there's hardly any oxygen left, it doesn't mean that it's completely dead. It just means that you wind up having a different type of life down there that thrives in anaerobic conditions. And again, a lot of these guys will use hydrogen sulfide. That tends to be a very, because there's so much sulfur in the crust, you see a lot of reduced forms of sulfur, and organisms have adapted to use that as an electron donor. And there are other organisms that can also live anaerobically that can feed off of those. A good example to try to drive this home is anyone familiar with the term meromictic? So a meromictic lake is one that never mixes. It never turns over. It's permanently stratified. So you have, in this case, what's called a chemocline. And this is a chemical gradient. And essentially, it's one where the oxygen is higher at the top, and it really starts decreasing as you go down further. And then you have another form, like hydrogen sulfide, that's present. And then it just becomes it's a transition between a different style of life. It's anaerobic at that point. And these that are never turned over, you'll see very prominent stratification layers of microbial life in them. So this is a lake in Russia, and you can see from the top here, this is the sampling apparatus that they have. It's sampling at different layers at this boundary where you have an oxic layer here and an anoxic layer there. but you can still get enough light that comes through this oxic layer where you'll have photosynthesis even down here, but you have anaerobic, well, not anaerobic, anoxygenic. You have photosynthesis that does not produce oxygen. And these are what are called the purple sulfur bacteria. So you'll actually see as you go down these just clear sections of water, then all of a sudden there'll be this huge purple film that's right at this layer. So this is where you have, these are your chemolithotrophs. They're utilizing the hydrogen sulfide that's coming up, that's being produced further down, and then utilizing that as an electron acceptor. I'll go into sulfur cycling in a bit, but still utilizing light as an energy source as well. So they're getting energy from light, but they're using an electron acceptor. They're using for reducing power to fix carbon, they're using H2S. I got ahead of myself. I actually have this planned out. But hydrogen sulfide is not the only thing they can use. There are different organisms that can also use, for example, ammonia. It's usually going to be in a bit lower concentrations down here because a lot of ammonia is oxidized in these upper layers. But there can be ammonia further down. Also, again, like in marine systems, reduced metals and even methane. The lower levels of these lakes can have, some of them actually have extremely high concentrations of carbon dioxide and methane. And this, this can be a combination of either, biological activity will produce a lot of methane and carbon dioxide, but there are some lakes that are meromictic that never mix at all, that they also get a lot of production from volcanic activity. Is anyone familiar with these African lakes, Lake Nyos in Kenya? I see a shaking head. There was some time ago where there was an earthquake and it disrupted that lower layer. And essentially what happened, it was like someone dropped a bottle of soda and then opened it. It just flooded out of this lake, and it's a large area lake. And all the carbon dioxide, and it's heavier than air, drifted out and flowed into the village and suffocated, I think it was some 1,300 people. So an entire village just died because they were suffocated. Most of that carbon dioxide was volcanic in origin, but there was still a lot of it that was produced by organisms that are feeding off of organic matter at the bottom of these lakes, and it builds up as well. So when it's not restratified, you get this buildup of these compounds that are further down in the lake. Let's do like a rapid transition and then move on to land. Soil ecosystems are quite different from aquatic ecosystems for obvious reasons, but I will go through that from a microbial point of view. So if we define soil, it's the loose outer material on the Earth's surface, right? And if we want to define it roughly, we can say that there are mineral soils and there are organic soils, but this is really a simplification. We can say that organic soils are from sedimentation of organic matter, which you find in bogs or forests. Mineral soils, really, it's the product of weathering of the parent material, so the bedrock that's below, and also alluviation, meaning that you have soils being, or rather particles transported in that build up over time from wind or water. That can also then be mixed in with organic material that is from any of the plants that are growing in the area, but you still have a lot of mineral material that's there. It's not just decaying or well-preserved organic matter. And mineral soils, of course, they can be defined by the percentage of different size particles. So you can have percentage of clay, sand, and silt. There's different systems for classifying size, and I mean, there's a whole science dedicated to just classification of different types of soil, pedology. So I'm not going to get into that. I'm going to keep it simple. But the idea here too is that you have a huge range of soil types, far more heterogeneous in space and time than water, than aquatic environments. And of course, in soils, you have air, water, and also living organisms that make up this matrix. And just to give you an idea of the spatial heterogeneity, if we do a pedology study and we try to classify soils in all these different forms, depending on particle size, but also the amount of organic material that's there as well, and a variety of other chemical properties. I mean, it gives you an idea when you look at a map of Europe of all the different soil types all over Europe. And just, you know, a sense of how heterogeneous it is. And you could actually go and zoom in on a small spot and you'll see a similar pattern because you can go further down into how you're classifying the soils. The pH might change over, soil pH can change even over the span of like a meter or less. And let me kind of dive into that a little bit, because when we talk about soil spatial heterogeneity, it's not just across the surface, it's also further down in the depth, the profile of the soil. So you have these different horizons of soil. So if you start at the top where most of the organic material is, that's from decaying plant material, in this upper area, this O horizon, this is usually where you're going to find the majority of life in soil. Really, you find, it can be anywhere from 80 to 90% to almost all of the life in soil is going to be in these upper layers, this A, this O and A, this topsoil. And as you go further down, you have these E and B horizons. So here it says loss of material. Of course, when it rains, you'll have organic matter that is being decomposed by microorganisms leaching in to this particular area. And you might get different levels of decomposition, or you'll get some organic material that is very difficult to decompose, and it remains there. And then as you get further down into the subsoil, you have essentially, like I said, leach material from the A horizon, and then you get further down into the C horizon, it's the weathered parent material, and then finally get into the bedrock. You can have microorganisms throughout the entire profile, of course, but majority of them, and majority of life is really going to be, I would say, in the top 25 centimeters. I didn't have a good diagram for that, but you can, when you map the abundances, you can easily watch how much they drop. I can show you that in another time. I feel kind of bad I didn't put that in. There's already a lot of material in here. But the thing about soil is that it's really composed more of soil aggregates. So an aggregate is essentially clumps of mineral material, organic material that are being held together by electrostatic charges, because clay is negatively charged. So when you have different minerals and you have different clay content, you get these clumps of soil that are called aggregates. And of course, the organic material from different compounds being produced by microorganisms, decaying organic matter, you get these clumps that are actually very difficult to break apart. And different soil types will have different ranges of aggregate sizes. But in general, the microorganisms, they can still inhabit every single part of that particular aggregate, but different soil types will have different pore sizes between them. So this is where you wind up getting the differences in whether or not they hold a lot of water after it rains. And that will control, yeah, there we go. They can, so microorganisms can, I'm getting ahead of myself, they'll colonize interior or the exterior of the aggregates. And so the exterior of the aggregates here, when you have water between these pore spaces, here is where you're going to have a fair amount of mineral or nutrient availability. Once you're inside, though, it becomes a very different environment. So to give an example of that, if you have a single aggregate, which might only be a half a centimeter in diameter, and you can even have smaller, you can have very substantial oxygen gradients from the center on out to the exterior of that aggregate. So on the inside, if you're an organism, you had better be used to living in anaerobic conditions, because the oxygen there is going to be borderline non-present. It'll be very anaerobic. But as you get further out, it'll be more normal conditions, more aerobic conditions. So you can get a gradient of different communities just across this small region. So this is why, and this is what you call a microhabitat. So really microorganisms have microhabitats, and this is especially true in soils. And it can become quite difficult to work as a microbial ecologist in soils because each aggregate might have a different community in it. So if you want to understand the properties of soil and the microbial communities that are in soil, you're kind of doing an averaging across all these particles. But when you start going down in scale, it'll change. And also from a microbial perspective, there's a lot of temporal heterogeneity. So over time, conditions are changing. I mean, a lot of this has to do when it rains. Of course, you're going to have less oxygen present in this pore space, because if it rains, there'll be organisms there that are going to be quite happy because there's water available. They'll start consuming oxygen, and the oxygen content will go down. It also pushes out the air, of course. So you wind up with anaerobic conditions when it rains. But then it starts to dry up. Maybe the nutrients become less available now because now you don't have these. Let me just go back up a little bit. You can see here, you might have water in some places, and maybe there you'll have a lot of dissolved nutrients, but here you won't. If you're a fungus, it's not such a big deal because you can span across all that. But if you're a bacteria, it might not be that good. So as a microorganism, a lot of these guys are adapted to rather fluctuating conditions because of the temporal heterogeneity, where you have, varying availabilities of oxygen and carbon and other nutrients that you need to build up your biomass. And because you have all these microhabitats and all these differences even across the aggregates, soil systems tend to be much more diverse than aquatic systems. There are, of course, always exceptions, but on average, there's this research project called the Earth Microbiome, which is actually quite interesting. And they've done studies or surveys of all these different habitats and all these different environments. And if you look at the y-axis, you can think of that as sort of the number of different types that they found, like species. It's a little bit more related to the molecular information, so it's like the DNA type. But you can see that as you go across different types of habitats, from animal corpses to the surface of animals to, here's plant rhizosphere, that's in soil. I'll get into that later. And you have aquatic, so you have host associated and free living. So if you look at aquatic environments, so water, aerosols, hypersaline, very salty, surface waters, non-saline surface waters, marine. You can see it's only when you get into sediment that you actually start to see a jump up in the diversity. But then sediment too is also going to be quite diverse, but soil really is even more diverse. And then when you get into plant rhizosphere, which is also in soil, it becomes even more diverse. So the other funny thing about soil, though, is that you might have all these different aggregates and a lot of surface area because they are packed aggregates. But really only 1% of that surface is going to be inhabited by microorganisms. So there's quite a bit of distance between each of them, like each individual cell. So interactions become a little bit more complicated in that regard between organisms, and how one organism might utilize a nutrient, produce another nutrient that another organism can use. A lot of that's going to be dependent on whether that produced nutrient can diffuse to the other organism. But because of the temporal variability, meaning that sometimes it might be just fine for growth, there's enough water, making nutrients available, and then other times it's going to be very difficult for growth, very harsh conditions. A lot of the cells, up to 95%, are going to be dormant. They're going to form spores or other structures where they're just not going to be, or the metabolic activity in the cell is going to be very low. And when you survey the different types or the different taxonomic groups, so the different groups of species that you can classify into families and orders and broader groups, a lot of these groups, like here you have the fungi at the top and the bacteria here, archaea, and then protists. Especially with the bacteria, a lot of these groups, like the acidobacteria, are in general noted for being highly tolerant of physical stresses. For example, they're called acidobacteria, you can probably guess, because they can tolerate low acid conditions. I don't mean like battery acid conditions, but things that are below a pH of maybe 5 or at a pH of 4. At the same time, they can also tolerate low water potentials. So if there's very little water, they can still be actually metabolically active, just at a much lower level. They won't be spores, they won't be dormant, but they'll just be sort of subsisting along. And also at lower nutrient levels. For example, acidobacteria, if you fertilize a soil, this group noticeably goes down in abundance. It's not adapted to when there's a lot of nitrogen that's readily available. It'll be outcompeted by other organisms, but it just doesn't like it. And the other thing with soils in particular, as you maybe saw with that other graph showing the tag types, association with plants is a rather critical factor in soils. Of course, it is in sort of these coastal areas where you have marshes and sort of kelp areas. But I'm going to kind of hop over that because we're also going to speak a little bit about microbes in agriculture. So transitioning from our soils, we can go a bit into how microbes in soils are associated with plants. So in this area, it's called the rhizosphere. So the rhizosphere can be defined essentially as the zone of soil that surrounds a plant root, where you really have, it's largely influenced by the actual plant itself, by the root. This can be, there's so many different definitions. I didn't want to give a specific one. I've heard the rhizosphere can only be defined because there's rhizosphere, then there's rhizoplane. Rhizosphere might be defined as like maybe the 3 millimeters of soil around a root. Other papers define it as the centimeter around the root. Other papers say, well, it depends on what kind of plant it is. So let's just say that it's the area that's influenced by the root most, compared to the bulk soil. And that's the soil that's, of course, much further away from the root, where you don't have as much, let's say, carbon compounds that are produced by the plant going into it. So it'll reach a limit once it diffuses out of the root. But that being said, it's not just carbon compounds being produced by the plant. You also have, this is the mucilage that roots produce in order to help it sort of glide through and make its way as it's growing in through the soil matrix. You have a lot of cells that are dying off, that are being sloughed off and being left in the soil matrix, but right next to the root. So all that becomes a rich source of carbon for different organisms, microorganisms. That being said, it's not just about the carbon around the root, but it's also the nitrogen levels, because in this area, yeah, maybe the microbes are perfectly happy that they're getting so much carbon, but the plant wants to use nitrogen as well, and so don't the microbes. So you get a competition between plants and microbes for nitrogen. And how they compete will define quite often the limits that the plant can grow to, and also defines any sort of strategies that the plants and the microbes evolve over time to get at that nitrogen. So for example, you can have these nodules that I'll get into that a little bit later, but you can have organisms that can fix nitrogen, that are going to be actually utilizing carbon that's coming from the plant root. These are the Rhizobium. I've read a couple of reports. They're very good. There's also, and those are bacteria, but there's also, of course, fungi. And AMF is, I'm going to go into that a little bit later too, arbuscular mycorrhizal fungi, where you have essentially hyphae that will infect and go into the cells, but it doesn't hurt the plant. It actually helps the plant sort of extend its root system in a way that, as it's providing carbon to the fungus, the fungi hyphae can then grow out even further and then take in nutrients from even further away in the soil matrix. So while it's doing that for the plant's providing with carbon, so it becomes a symbiotic relationship. But that being said, I wanted to first go into sort of the generals behind biogeochemical cycling, because this is really what's driving, I guess the best way to put it, it's really how you can better understand the relationship between microorganisms and the habitat that they're living in, because the organisms are taking basic elements, trying to either incorporate them into biomass or utilize them as energy. And then those elements are no longer available in this large inorganic pool. But then the organisms die, and then you have a cycling back into an abiotic pool. So the idea of a biogeochemical cycle is you have immobilization, where they might be available in mineral form, being utilized by the organisms and made immobile. So it's no longer available to other organisms. That organism's taken that, and it's now its own. But when it dies, or when it's doing other particular processes, you have mineralization. So then it goes back into inorganic forms. But the thing is, you can look at it from an ecosystem perspective. So carbon, it can be in abiotic forms, carbon dioxide. It's then immobilized as macromolecules, and then it's mineralized by heterotrophs back to carbon dioxide. But from the organism perspective, you have compounds that are being oxidized, and then you have compounds that are being reduced. And I think I got that backwards. Oh my goodness. No. Yep. So a reduced is when it gets electrons, and oxidizes when it loses them. Hey, cool. Whew, I was worried I had that backwards. But the idea being is that as you have electrons flowing from one place to another, that is energy. It's also a way in which you wind up building up or breaking down carbon compounds in order to either build up your biomass, or if you're breaking them down, you're gaining energy. Or you can be utilizing them for other processes, but but by and large, you're gaining energy from that breakdown. So you're building biomass. Or if you remember this is our redox tower, right? So you have compounds that are electron rich, they're reduced, and those that want to suck up electrons, they're highly electronegative. And the difference between how electropositive or electronegative they are, that gives you an idea how much energy you can get when you couple those two together in a metabolic pathway. So maybe we can start off with carbon cycling. Should be rather self-evident. Carbon is the bulk of living organisms. And this is always interesting to think about. These, anyone familiar with these Redfield ratios? He was a marine biologist, Redfield, and he wanted to study what was called the stoichiometry of elements in the ocean. So what are the ratios of elements like carbon to nitrogen to phosphorus? I mean, these are some of the most important and abundant elements in living organisms. So by sampling in different areas and then just basically measuring the, quantifying, you get this average of, in oceans, for example, 106 parts of carbon to 16 of nitrogen to 1 to phosphorus. And you can use that ratio to get an idea on what kind of habitat you might have, because as that ratio might change a little bit, you can see whether one particular element is going to be more limiting to growth than another. So for example, in soil, overall, it's different from oceans. You have 186 to 13 to 1. So you have much, much, much more carbon. But a lot of that has to do with the fact that the microorganisms that are living in it, you see that now you're looking at soil microbes, it's 60 to 7 to 1. So that means there's a whole lot more nitrogen and phosphorus that's being incorporated into the microorganisms. So that gives you an idea of what might be limiting in the soil environment. So nitrogen is quite often, and phosphorus are quite often limiting. That's why we fertilize, right? For agriculture. But in carbon cycling, I think the best way to put it is you can really break it down. You have two gaseous forms of carbon. You have carbon dioxide and methane, right? But you have two essential processes that are driving carbon cycling. You have carbon fixation and decomposition. So the primary production, where you're taking in carbon dioxide, converting it to large macromolecules, and that's being done by photosynthesizers. I mean, this is plants too, but in this case, we're thinking of not only plants and not only photosynthesizers, but autotrophs that utilize chemical energy. So these chemolithoautotrophs. So they don't need light, but they can still utilize other minerals to get the energy. They will also fix carbon. And they will do that under anaerobic conditions or aerobic conditions. But then, of course, you have decomposition, where that's being performed by the heterotrophs. So they're able to consume the carbon molecules that are being produced by the primary producers. And to get energy, they take that, break it back down into carbon dioxide. So you have this cycle between how the carbon's being utilized by two different groups and two different processes of organisms. And I mean, you can have both of you can have these processes going on in either aerobic or anaerobic conditions. But methane is a bit different. It's, I would say, a little lesser part of the overall carbon cycle, but it can be quite important because methanogenesis, so the process of producing methane, it's really one of the final steps in anaerobic decomposition. If you remember that film that I did, where I went out into the swamp and then was able to collect all the methane and then light it on fire. What was happening, and I explain this, actually, this kind of goes back to what Folkmar was talking about in his lecture, where you have these fermentation processes. Because this is occurring anaerobically, complex carbohydrates are being broken down into simpler carbohydrates, and then you get fermentation by primary fermenters, and then you get these very simple carbon compounds, 2 carbons or 3 carbons, and then you can have different processes that in the end result in the production of methane and carbon dioxide. So you can have acetogenesis, and methanogenesis that just produce, or you have these syntrophic reactions where you have two species of organisms that work together to make an otherwise difficult reaction possible just by the thermodynamics. But at the same time, you have methane being produced, you can also have methanotrophy. So the methane can then be utilized by other types of organisms, and this can occur aerobically, and recently they're speculating that it occurs anaerobically as well, and that's actually fairly recently discovered, where you can utilize the methane both as an electron donor, and you can utilize it as a start for biosynthesis. So you can use it for energy and for carbon, for building up biomass. So how these two processes interact, as you can imagine, they're extremely critical in environments like melting permafrost, where if you remember, you had these, you can classify soil based on organic or mineral, and a lot of permafrost soils, they're very highly organic. There's a lot of basically non-degraded organic matter in these environments. And I don't know if you've seen this, but there's a good example of thawing permafrost. It's called the Batagay megaslump. This is in Siberia. And it's been growing. It's what you call thermokarst. So it started off as a small gully of drainage water because the region was deforested and a bit abused. But as things have been warming, the depression has been getting bigger and bigger over time. And this gives you an idea how big it is if you see the little guy here. It's enormous, and it's growing. I forget, it's like 50 meters every year, I think, along those lines is what the figure was. But when you start having these thawing events, you have giant plumes of methane that are also going to be being produced. Maybe not necessarily, I mean, you'll have it being produced from slumps like this because you've disrupted the whole system. So when it's no longer stratified, methane, first of all, it's warming up. So these organisms that do methanogenesis are capable of, they're more active. But then also when things are less stratified, you can wind up having a larger production. This is what they're finding in these melt ponds as well, up in Arctic systems. There's actually a researcher in this department who studies what are called pulsas. And they're these upwellings of thawing permafrost that are just starting to thaw, and they become very, they're very high producers of methane. But the interesting thing is, do you wind up seeing a concurrent growth of methanotrophs? And what environments do they live in? And what conditions do they prefer? So how those two different groups interact can also have a big influence on methane production. So, oh boy, we're gonna go over. So with carbon cycling, I can just say that on land, most of the carbon is going to be in decaying matter that's in soil, so this top layer of soil. And then in marine systems, a lot of it is dissolved CO2. So that gives you, these numbers here give you an idea as to the proportions of global carbon that are in the different environments. So the oceans are a huge sink of carbon. But it is also nerve-wracking to know that permafrost has a very, very high amount of carbon that is just sitting there and not currently being, well, it's a large storage of carbon. So that's why there's a large concern that as you have increased warming, you're going to have increased methanogenesis and increased breakdown of this carbon. And then you might have much higher rates of greenhouse gas emissions from these environments. So moving on to nitrogen cycling. So atmospheric nitrogen is the largest pool, as we probably well know. It's 78% of the atmosphere. But the thing about nitrogen is that it's completely inaccessible in that form to life. So it has to be converted to what we will call reactive nitrogen before it can be used by organisms. And this can be done by lightning, abiotically, and that's really the only way. So you'll have lightning that will produce an oxide of nitrogen that can then react and form different forms of nitrogen oxides. But most, and that was maybe done on very early in Earth's geological history. But for the most part, organisms have evolved to perform enzymatic fixation of nitrogen. So this is really the main way that nitrogen has entered into the biosphere over geological time. And the thing about it is it's very expensive. It takes quite a lot of ATP to fix a molecule of nitrogen. So the organisms, this is not something that they're doing and having a nice time doing it. They're doing it because they really need the nitrogen. So it's very energetically expensive. But after nitrogen fixation, you can have, and when I say reactive nitrogen, you can have these different forms that can either be ammonia, nitrite, or nitrate. And you have a variety of different processes that convert from more reduced forms, which is ammonia, to more oxidized forms. I mean, in addition, you can have ammonia being synthesized into biomolecules, nitrate as well. So you can go in that direction. You can also have respiration processes. So the organisms, rather than using the nitrogen to make biomolecules, they're respiring it. So you have nitrification, where they're using ammonia as a donor to get electrons. Or you have respiration, where they're using the nitrate as an electron acceptor, like we use oxygen as an electron acceptor. And there's another process called a dissimilatory or respiratory ammonification. I won't go into that too much. But the idea is that eventually, after you cycle through these different forms of nitrogen, it does get returned back into the atmosphere in a couple different forms. It can be denitrified completely or converted back to nitrogen gas through denitrification, or another process called anaerobic ammonia oxidation, which is it's a very interesting pathway, but not enough time to get into it. But I'm going to come back to nitrogen cycling in a little bit here. And just one last thing to go through is sulfur cycling. So one of the things, and one of the reasons I kept pointing out hydrogen sulfide, is that sulfur is a rather abundant element in Earth's crust. And predominantly you find it as calcium sulfate. You guys might have recognized that one from what you used in some of your Winogradsky columns. So gypsum, yips. You also find it in iron minerals and pyrite, and it can occur elemental forms as well. The oceans really are the largest reservoir of sulfur, in the form of sulfate. And for those of you that live close to the ocean, you might recognize this, because marine organisms will utilize the sulfur, because sulfur can be incorporated into biomolecules, not only proteins, but also organic compounds that can control the osmotic stress of the cell. So how easily water can go in and out of the cell. A lot of algae produce dimethyl sulfide as a means, or it's actually dimethyl sulfopropionate, but then it gets broken down into dimethyl sulfide. So when you're walking by and you have this sulfur smell of the sea, this is what it's coming from. These algae and the compounds that are breaking down. You can also have sulfur in the ocean can also control cloud formation. So when the dimethyl sulfide becomes volatile and goes into the atmosphere, you can actually have, in addition to other factors, you can get these sulfur molecules in the atmosphere that wind up causing enucleation of clouds. So the sulfur cycle actually controls also weather cycles in oceanic systems. And just to give a nice example of sulfur cycling, because you can have, if you go back here real quick, so you have processes where it's being oxidized, which can occur aerobically or anaerobically, and it's being reduced. And there are really interesting, oh, and by the way, of course, you have assimilation into proteins as well. But you have these interesting different ecosystems where, in addition to like the lakes, the meromictic lakes that have these layers of aerobic and anaerobic, where you have the photosynthesis going on, anoxygenic, you can have organisms that are capable of utilizing hydrogen sulfide, converting it to sulfate, living very close to organisms that can utilize sulfate as an electron acceptor and converting it back into hydrogen sulfide. So these are actually called pink berry consortiums. And if you go in the sediment of this particular area in the States, this is actually from, that's from Massachusetts, and you dig up the sediment around the plants, you see these little tiny pink berries that are full of two different types of bacteria, one of which is photosynthesizing. So it's utilizing sulfur, and it's, well, yeah, from the sediment. So here we go, HS, so it'd be H2S. It's reducing that, and it's utilizing carbon dioxide, producing carbon that can then be used by another organism. But that organism, they're still living in an anaerobic environment. So then it utilizes sulfate, which is the byproduct from this organism, as an electron acceptor, producing hydrogen sulfide. So you get this cycle just within this little berry of a closed ecosystem, or a closed cycling of sulfur. And then, of course, it's utilizing carbon dioxide, or rather carbon, produced by this primary producer. So you get these interesting sulfur cycle habitats. Yeah, let's move on. Whoa, oh yeah, one last thing. This is really cool. There are some organisms too, because as I pointed out, you can have sulfur cycling occurring in anaerobic or aerobic conditions. But the thing is, you're going to find most of hydrogen sulfide occurring in anaerobic conditions, but you still want to have an electron acceptor. So the sulfide is the donor. So it's taking in the electrons and it's acting as a battery. It's going to something else as the acceptor, and then that's what's generating the energy in the organism. There are some cells, there are some organisms that have actually developed a lifestyle where they make filaments that extend through the sediment. And they find these in marine or lake systems, and they can extend up to a centimeter. This is 2,000 times the length of the cell. And it becomes an electrical wire, because what they'll do is they'll take the electrons from the hydrogen sulfide, transport them up into in the anaerobic area, transport them up to where the oxygen is available, and then that's what's actually generating the proton motive force that gives the energy for the organism to live. So rather than finding a different electron acceptor in this area, it transports it up into the oxygen. So that becomes a form of aerobic respiration, and that typically gives you more energy because there's a bigger difference, right? There's a bigger electronegative difference between the oxygen and the hydrogen sulfide. You see, if you compare that to where iron or manganese or other potential acceptors are, this is lower energy, but you go up to oxygen, you get more energy. So they've evolved a way to maximize that. And you get electric wires made out of bacteria. And you can see that you have these very specialized structures that they produce that are ridged around the bacteria that just allow for the flow of electrons to go up through these ridges into the sediment in the upper part of it. So there are living electrical cables. I just find that really cool. There's multiple other biogeochemical cycles I could go through, but we're already going over time. One that's being studied more currently now is mercury, because you can have mercury being essentially forming different compounds, making it more or less available to different organisms. So it can transport its way through the food chain mainly through the activity of microorganisms. So because it's changing the charge on the mercury atom, adding or subtracting electrons from it. Okay, and finally, we can talk a bit about microbes in sustainable agriculture. And it's a good transition from biogeochemical cycling, because really, when we're working in soil systems and we're working with crop production, we're trying to manipulate these biogeochemical cycles to maximize the nutrients that are available to the plants, in addition to trying to keep the plants healthy in their capacity to fight off diseases, to produce higher yields of different kinds of whatever it is that they're producing for the food product that we want. And like I said before, I'm going to leave the part about rumens and animals for next week. But when we talk about nutrient cycling, I'm going to go back to nitrogen because A, this is what I work with, and B, I think it's a more pertinent cycle to go through, because nitrogen is one of the toughest ones to get into the crops because it's so difficult to fix, meaning that all of it is available, most of it's available as atmospheric nitrogen, and it's not available to the plants. So you need to have some way to get the nitrogen into the plants. Otherwise, you wind up with very poor yields, very poor, for example, in corn, you can see nitrogen deficiency winds up giving you very poor ears of corn. And it's easy to see the effect when you have areas of low nitrogen to high nitrogen. You can easily see what happens and the difference between being fertilized and not fertilized with nitrogen. Historically, we relied on nature to provide the nitrogen. So it was through fixation, it was through the utilization of manures. And actually in the 1800s, before industrial processes, there was actually the what's called the guano craze. So you actually had people going out into the middle of the oceans to hunt down islands that were just enormous collections of bird guano, just bird poop, which is mostly urea, uric acid, right? And it was so lucrative that the US government had what was called the Guano Islands Act in, I think it was 1840, which meant that any American could go out into the ocean, find an island and say, this is American now, because it had guano. And there's a lot of islands in the Pacific that are still American territory because of that. There's nothing on them, just birds and bird shit, but they still belong to America. However, this changed as we had the invention of the Haber-Bosch process. And this is probably familiar. Ooh, I spelled that wrong, didn't I? Anywho's, this is probably familiar to many. So this was the process by which you could then fix nitrogen using a chemical process, primarily metal catalysts and pressure and heat. So this gave rise to the large chemical fertilizer companies that we see nowadays. But, you know, there's a negative side to that. But the thing is, it fed a lot of people. You saw crop yields expanding dramatically during the Green Revolution through the use of nitrogen fertilizers. A good example is given here, and this is just a few countries, but you can see over the period between 1961 and 2001, if you look at the yield, and I think this is tons, I covered that up, I just want to give a general idea. You can see that the amount of fertilizer that's being used over time from 1961 down here up into the 2000s. It mixes around a bit, but you get increased yields along with that usage. So this has helped a lot of people, billions of people, not starve to death. Was the invention of how to get nitrogen cheaply into crops. So that's why I wanted to stick with just this one, nitrogen. But the issue is that with that, you have a massive increase of the amount of N, reactive N, so ammonia, nitrate, that is going into the environment. So over time, you get a gradual increase in nitrogen that's just coming from biological fixation, but you can see how disproportionate it is to the amount of fertilizer nitrogen that's going into the ecosystems. And of course, this is all in line with the start of the Green Revolution. But something to really consider is that the amount that we're fixing is approaching the amount that's fixed globally every year. There's terrestrial, because we're fixing it and putting it onto land. So we're fixing it for terrestrial use. But there's a massive amount that, of course, is fixed in oceanic systems. And we're actually starting to approach that. So you might have heard of these ideas of these planetary boundaries. And nitrogen, the amount that we're putting in is approaching the boundary where we might not be able to put in more nitrogen, because it might throw everything out of balance. And I'll get into that in a minute here. So we have a massive increase of nitrogen into the biosphere. And that, of course, leads to eutrophication. But a big factor too, and this is what I work with, is that you have increased emissions of nitrous oxide, which is a very potent greenhouse gas. If we look at all the greenhouse gases, the major ones, nitrous oxide is 8% of that. So we have carbon dioxide, you have methane, you have nitrous oxide, and I think you have water vapor. And then there's a few other smaller ones. But the thing about nitrous oxide is that it's 300 times more potent than CO2 as a warming gas. So, and it is also the main cause of ozone depletion. So if you remember the chlorofluorocarbons back from the 80s, and they were going up and destroying ozone, now that those have been regulated, ozone destruction is mainly caused by nitrous oxide. And this has come as an unfortunate surprise, because it really stems from our use of fertilizers. So you can see that you have, as you increase the amount of fertilizer that's being used, you get in all different areas higher rates of N2O emitted from the soil. And this is from two microbial processes, mainly nitrification. So that's going from ammonia here over to nitrate. This is aerobic, and denitrification. So we have nitrate under anaerobic conditions being converted back into nitrogen gas, and that's emitted in the atmosphere. But the thing about denitrification and nitrification is you can get nitrous oxide being produced by both processes. And that gets increased the more nitrogen you add into the system. So when we want to work, oh, and here's a good figure showing this. So if you look at sort of the overall fluxes of nitrous oxide, so the amount that's being emitted into the atmosphere and the amount that sinks, because ozone is actually the main sink of nitrous oxide. Agriculture, as far as anthropogenic sources, is by far the largest contributor. So if we want, and if we have continuing increases in soil N2O flux, we might be able to get our carbon emissions down, but it will be offset by the amount of nitrous oxide that we're producing. So it doesn't matter how much carbon footprint you work with. If we don't take care of the nitrous oxide, we could also have a big problem with greenhouse gases. So this is something that's very much in we're trying to understand how we can do that, how we can manage crops to reduce the amount of nitrogen we're putting in. And really what we have to do is figure out how do we control the microbes that, or rather at least manipulate them and understand their ecology better, that are controlling these different processes. So to put it really simply, you have processes that will allow for nitrogen to be retained in the soil. And that comes from nitrogen fixation. Of course, it puts in nitrogen. But there are also processes that make sure that nitrogen stays in the form of ammonia. Ammonia is positively charged, so it sticks in the soil, because clay particles are negatively charged, so they'll bind better. The problem is when you have things like nitrification, which is taking ammonia and converting it into these soluble forms, that can get leached out, and then you have nitrogen loss. In addition, you'll have nitrogen, or nitrate, that will then be used as a substrate for denitrification under anaerobic conditions. And this is what produces the bulk of nitrous oxide, is denitrification. So if we can control how the balance going between these different processes and the microbes and their ecology, we might be able to better manage nitrogen so we don't have such large problems. One way that people are looking into this is actually with something called biological nitrogen or nitrification inhibition. So again, nitrification, we're going from, I went like that, sorry. We're going from ammonia, it's being oxidized and converted into nitrate. This we want to avoid. We might be able to do that if we find plants that produce specific compounds that actually inhibit the activity of these organisms. Therefore, the nitrogen stays as ammonia, it can be utilized by the plants, and it also stays in the soil system. So rather than being leached out or denitrified into N2O, it gets retained in the biomass. And there are different grass species that are being sort of put into crop rotations, where you can see that when you have a control versus just soybean versus adding in different types of grasses, the ammonia oxidation rates go down and the cumulative N2O goes down, all because nitrification is being inhibited by these plants. So the organisms, these are archaea and bacteria, ammonia oxidizers, they don't oxidize ammonia anymore due to these compounds being produced by the plants. And this is important to think about as well, because the other trend that's a bit disturbing is that different countries will have different nitrogen use efficiencies. A good case, and this is not to bash China or anything like that, but this is a study produced a few years back where they're looking at the amount of fertilizer being applied versus the amount of yield of a crop. I forget what crop it was. I think it was wheat. So the yield that you get per amount of fertilizer application rate. So here, since 1961, you've had this trend where you've had nitrogen fertilizer in the US being applied at increasing rates, but it sort of tapers off the amount being applied, but you still get higher yields through the development of new cultivars of plants that utilize nitrogen more efficiently and different agricultural practices. But in China, it's a bit different because you simply have increases in the nitrogen being applied, and it increases, but you don't see an increase in yield. And a lot of this has to do with the efficiency. So yes, of course, there are different cash crops in different countries, but when you measure just the amount of nitrogen being utilized per amount of crop, or rather the amount of used by the crop per amount applied, you have a much different perspective in the US versus in China. So you have a lot of nitrogen that's just being wasted and thrown into the environment when you have very low nitrogen use efficiency. So that means maybe fertilization practices have to change. And how do we put nitrogen into the soil in a way that's more sustainable? Naturally, we look towards nitrogen fixation. We go back to how can we enhance this process and how can we utilize it, but still see the same crop yields that we see in order to feed a growing population. So nitrogen fixation in agriculture, for those that this must look familiar to, I know several of you. So you can have nitrogen fixation occurring as rhizobium in symbiosis with plants and legumes. So you'll have the start of it. We have root hairs that get infected by a rhizobial cell. And then you get this, what's called an infection thread. After the cell recognizes that, oh, okay, you have these particular chemical factors, I'm going to grow this way, in a certain way, and start growing more and more. And then eventually, after the bacteria forms these nodules where you have the thread that goes in, the cells start to grow more and more due to hormones being produced by the rhizobium. And then you get nodules that are become, they allow the bacterium to fix nitrogen because they provide the right conditions. And the plant is able to provide carbon to the bacterium, and the bacterium is able to sit in this nice little nodule and then, produce nice little compounds that get rid of oxygen, because nitrogen fixation cannot occur when oxygen is present. The enzyme is completely destroyed. So you have these nice little globins that go and suck up all the oxygen, and then the bacterium starts producing nitrogen that the plant and the bacterium can then use. But that's not the only way. You have free-living nitrogen fixation. And this is a diverse range. And these are just organisms that are living freely in the soil. And it's a diverse range of different species, some of which are aerobic or anaerobic, and some of which are heterotrophic. So they're using carbon for, just basic heterotrophic metabolism. Or there's like cyanobacteria, and they're photosynthetic, but they also fix nitrogen. And just to give an idea of the amount of nitrogen you can put into the system, depending on the crop that you use, which will also determine the species of rhizobium that you have, and how you manage it, you can get different amounts. And this is kilograms per hectare per year. And if you go back real quick, just to give you an idea. So here we have kilograms per hectare per year. So the fertilization rates in the States here that topped off at 150 kilograms per hectare per year. And you can see the amount of nitrogen you can fix becomes comparable to that. However, you have to balance that with what the plant is using too, because it's not like all that nitrogen is going right into the soil. The plant's using it. So how you manage the plants, i.e., do you harvest them completely, or do you till them back into the soil to maintain the nitrogen there, determines what you can do with using nitrogen fixation for managing nitrogen. And last but not least, about nitrogen fixation, it's inhibited by nitrogen fertilization. So if there's enough nitrogen present, you won't get any fixation. And this can be actually a bit worrying, because if you have fields that have been fertilized over long periods of time, you can actually see a decrease in the number of nitrogen fixers that are there because they're being selected against. So you start losing that function in your soil just because you've been throwing in nitrogen for the past 40 years. So transitioning to an organic system can be a bit problematic. It's a concern. I'm only going to say something quick about the other type of symbiont that could be really useful in mycorrhizae. So you have, again, these fungal species that can form these globules inside the plant cell, but it's not hurting the plant. It's actually providing the plant, if you look to the right here, the left, right, it can actually act as an extension of the entire root system for the plant. So it allows the plant, when conditions are stressful, to go further out to get water and nutrients, while at the same time the plant is providing carbon and other nutrients for the fungus to survive. So you get this symbiotic relationship. This is particularly important in forest ecosystems, but it's equally quite important in any agricultural arable crop system. And you can have, this is actually something that was in our department, Roger Finlay, senior professor. You can have interactions between bacterial species and mycorrhizal species in this area, the mycorrhizosphere, right? Mycorrhizal sphere. Ew, hard to say. The idea being is that when you have both types of organisms present, they can complement each other. So for example, mycorrhizae are known to enhance nodulation by nitrogen fixing bacteria in plant roots. Mycorrhizae can also ward off plant pathogens by producing a variety of different compounds and also eliciting the plant stress response that makes it more difficult for a pathogen to infect it. So when the mycorrhizal infects it, the plant can wind up producing different compounds in a different response that the pathogens don't like, so they avoid it. And the last thing I'll add, you can also have, not just when it comes to nutrients, but also when it comes to plant stress. You can have a variety of different interactions between microorganisms and plant roots that will allow it to survive a variety of different physical stresses, like drought or like increased temperature. This was in one of the DeVries article that I sent, and it just shows different possibilities of interactions as you have different types of drought conditions. So for example, you might have happy conditions where you get mycorrhizal fungi spreading out into the soil, taking in nutrients for the plant. Also, you have other types of bacteria, and that's one thing I should add. Some bacteria, not only can they fix nitrogen, but there are other types of bacteria, many, they actually produce hormones that the plant recognizes and will enhance the growth of the plant. There's one hormone called auxin. Auxin causes elongation of things like shoots and also roots. And some bacteria, and these hormones, they're produced by the plant in different growth stages. But some bacteria also produce these compounds. And they will also make the plant grow in addition to what the plant is producing itself. So you get these plant growth promoting bacteria species. And I'm not naming all the species because there's lots of different ones, and the names aren't so meaningful as much as what they can do. But when we go back to the stress response, how the plant reacts to the stress will also determine what the microorganisms are doing and how well the system can recover later from that stress, from the drought stress. So if it's really becoming drier, the plant's not going to want to be putting down so much carbon. It's going to slow down its water flow into the roots, or rather the sugar flow into the roots, because the overall metabolism is going to slow down, try to conserve as much water and energy as it can, shut down the stoma, and then you wind up with a different interaction with the microorganisms. And eventually, if you really have extreme drought, everything just dies. But you have recovery, and after that connection between the organism and the plant is broken, you might have a completely different community that's inhabiting the plant later on, because the carbon is gone. Organisms that were feeding off of that carbon have probably died out, but there could still be other organisms that are very, very stress tolerant, and they wind up taking over. And then when conditions change back again, they're still around, and they might make it harder for other organisms to come back. So you get a shift. But when it comes to protecting the plant, you can have organisms, microorganisms that are producing other types of hormones, just to try to keep the whole system functional. For example, you have, here's just a few examples. In heat, you can have fungi that are, that actually it's a combination of a fungi and a virus that it infects the roots. It can actually increase the plant's heat tolerance. You have cold tolerant bacterium that are able to fix nitrogen when no other, and these are free living, these Pseudomonas. They don't form nodules, they just live out in the soil. But if it's too cold to fix nitrogen, but the plant is still able to grow, these guys can still fix nitrogen. So they help the plant when it's cold stress. And then of course you have drought, where the mycorrhizae can spread further out and get water from further away. Like I said, maybe if it's extreme drought, everything just dies off. But mild drought, you can at least have it extending further out into the matrix, drawing up from a greater area enough water for the plant to survive. And there's a few other examples for when it comes to, like, you have organisms that can be antagonistic to pathogens. So they might be viruses, or they might be bacteria that actually produce antibiotics or other compounds in the rhizosphere. Some that produce certain types of compounds that are osmotic protectants. So like this Bacillus, it's interesting because Bacillus thuringiensis is also an insectivore. This will actually, you know, the BT corn, the modified corn, it's from this bacterium that they get the gene that actually kills off the insect. So it has other functions too. And it's a really common soil bacterium. Yeah, and many other examples. These are just a few that are listed. So